Hello and welcome to the AP Bio Review video series. This is chapter 12. We're going to talk about the chromosome basis of inheritance. Okay, so you have to understand how the chromosome theory of inheritance connects to the physical movement of chromosomes in meiosis, to Mendel's laws of inheritance. And this is one of those test questions you guys didn't do real well on, so we're going to look at that diagram pretty closely here. Okay, so here in figure 12.2, we're going through this process of meiosis. Oops, I'm moving the whole slide. Hold on. Get the right tool here. We're looking at this process of meiosis. And in meiosis, we have meiosis 1 here, and then they kind of jump through the steps because we don't really need to look at all of them. And then we get into meiosis 2, which is down here. And so we're going to remember, when we're dealing in meiosis 1, the ants go marching 2 by 2, remember that? And then when we're dealing in meiosis 2, the ants go marching 1 by 1. And so if you get confused about your images, here's our split between meiosis 1 and meiosis 2. Okay. And so essentially what's happening in meiosis 1 is we call meiosis 1 our reductional division. And that is because we're going from 2N to 1N. And that happens when those homologous chromosomes, those homologous pairs, separate. And so the homologous pairs lined up on the metaphase plate, and then our microtubules, our kinetochore microtubules attach, and then they pull apart those homologous chromosomes. Then it's not until meiosis 2 that we're actually splitting up the sister chromatids of each individual chromosomes that were lined up on that metaphase plate in meiosis 1. So when we look at this, there's two of Mendel's laws. We have the law of segregation and we have the law of independent assortment. The law of segregation is saying 2 alleles for each gene separate. Well, those alleles are on the 2 different chromosomes. And so that's what this is showing you right here. Okay, so that is a part of meiosis 1. Over here, it says the law of independent assortment. Alleles of genes on non-homologous chromosomes assort independently. So what that is saying, and I'm going to erase some of this here. Let's erase all of this. What that is saying is that how chromosomes are lining up on the metaphase plate, how they're doing that is completely independent of how any other pair of homologs is also lining up on that metaphase plate. So in this image, we have the maternal chromosome being red and the paternal being blue. We have 1 red and 1 blue on each side of that metaphase plate. Over here, we have 2 reds and 2 blues on each side of that metaphase plate. And each of those options are equally likely. And because each of those options are equally likely, when we start looking at organisms with a large number of chromosomes, the number of different gametes that can be formed is absolutely huge. And we did the math back in Unit 2 when we were learning about cells. But the whole idea behind the law of segregation and the law of independent assortment is just to say all of these things come apart when we go through the process of forming gametes. That's what meiosis is doing. And how they come apart is independent of how everybody else is coming apart. And the reason this is so important is because it allows statistics to work. Remember, with statistics, when we're dealing with probability, we're dealing with independent events. Well, if we didn't have the law of independent assortment, we wouldn't be dealing with independent events. And that's what's going to happen a little bit later on when we start trying to figure out linkage and those sorts of things. So all of this stuff that's happening with Mendel is actually part of meiosis 1. So all of his laws are coming out of meiosis 1. Meiosis 2 goes on to actually form those gametes, but it's meiosis 1 where those laws from Mendel are actually coming into play. Okay, we need to know the patterns associated with sex-linked genes. So we're going to remember that the way we figured out sex linkage in the first place had to do with flies. And the reason it was so peculiar was because Morgan found that white-eyed fly, and that was a mutant. And he knew it because he'd never seen one before through all of the generations of flies he'd worked with in the past. And it happened to be male. But he had this one precious white-eyed fly, and after spending his whole career looking at flies, he didn't know really what to do with it, but to breed it with others to try to get more white-eyed flies. They're flies. They don't live real long, so you have a short period of time. So he took his white-eyed fly, and he was mating it with all of these females, and because all he had in his lab at that point were red-eyed flies, he kept getting white-eyed flies and more red-eyed flies and more red-eyed flies and more red-eyed flies. And he's very frustrated by this because he had this one white-eyed fly, but he couldn't seem to get any more wide-eyed flies out of it. Well, it wasn't until the progeny here started having their own babies, because, they're flies, they live in a vial. It doesn't matter that they're all brothers and sisters, they all have more babies. And it wasn't until that happened that he started to get more of his wide-eyed flies. And lo and behold, all of the wide-eyed flies were males. And that is how we came to understand the concept of sex linkage, that there could be genes that followed particular sexes, and there are genes that are attached to particular chromosomes, and that was causing some unusual patterns to kind of appear when we started looking at these Punnett squares. And so in this case, your wide-eyed flies here were happening because they have a Y chromosome, not altogether too different from humans, but there was this Y chromosome that was taking the spot for one of the homologs in that homologous pair. And then we had the X chromosome, and that's where that wide-eyed trait was actually being housed. And so the reason he wasn't getting any wide-eyed flies up top is because mom up here had red on both of her X's, and so she was always giving that red, that wild type red, to all of the progeny. And that was turning out to be a dominant condition, and then the presence of one wild type red allele was kind of masking that white allele in the females. But that the trick to knowing that there's some kind of sex linkage going on is to look for very unusual patterns with regard to progeny. Anytime you see males and females being split out in your data and the numbers being vastly different, that's how you know that you have some kind of sex linkage going on there. So here are just the Punnett squares that are associated with sex linkage, and in this case, what we're looking at is an affected male, and then a homozygous dominant female. And the way this diagram is depicted is to show this is affected, this dark color. This medium color, those are going to be your carriers. So they are phenotypically normal, but they are carrying one of those deleterious alleles. And so when we do this, when we look at these, here are your parent generations. And so out of this is going to be your F1 generation. Now, the way this is set up, notice that this is A, B, and C. These are not F2s down here below. They're just showing you different combinations of sex linkage. So even if a parent has an affliction, it's possible that none of the kids will. And conversely, here neither parent is phenotypically had this condition, and yet they can have children who are affected by this condition. And in this case, the carrier is mom and dad is phenotypically normal. And so because mom is a carrier, half the time she's going to give this affected condition to her kids, which is fine for her daughters because dad's going to give a normal allele to the daughters. But when dad gives that Y to the sons, that's when we run into this issue of having affected sons. So that's how both parents can be phenotypically normal but have an affected child. Similarly, or I guess conversely, it's possible for one parent to be affected, one parent to be a carrier, and then to have some kids that are neither. And in this case, if dad's giving that Y, which has no trace of an affliction because it's the Y, and mom is giving the normal allele, that child is going to come out without the condition and without carrying any of it. And so in contrast down here, these are your affected children because we have 2 copies of that affected allele in the girls and 1 in the boys. Remember that the term for this in the boys is hemizygous. Okay, and this is hemizygous is only true for boys with regard to these sex chromosomes, and it's because they only have 1 copy of the X, and so that 1 copy is what's going to be expressed regardless of any other inheritance. And so it doesn't matter if it's dominant or it's recessive. He's hemizygous; he's going to express it. You have to know how the alteration of chromosome number or structurally altered chromosomes can cause genetic disorders. All right, so we looked at some non-disjunction diagrams in class, and we just talked about non-disjunction means that the chromosomes aren't splitting up the way they're supposed to. And that can either be in meiosis 1, right, where we're supposed to divide up our homologous pairs, or it could be in meiosis 2, where we're not splitting up our sister chromatids. The results aren't really all that different aside from how many gametes are affected. If you have an error up here in meiosis 1, none of the gametes are normal. If you don't have an error until mitosis 2 will have 2 normal gametes and 2 abnormal gametes. And so that non-disjunction is just saying, disjunction is them coming apart, so they're just non-disjunction is you didn't come apart, and if you don't come apart the way you're supposed to, moving forward, there's going to be some kind of errors in meiosis. This is how we end up with something like trisomy 21. So other major changes in chromosomes that could cause some issues are different rearrangements, if you will. So we can have a deletion where we quite literally delete. I love the names of these. They just make sense. Duplication means that we have too much of stuff. This is what we saw with the ice fish example. We had duplications. Inversions, if you invert something, you flip it around. And then translocation is when 2 parts of chromosomes swap spots. It's kind of like a crossover event, but it's a mistake because the crossover event is supposed to happen between 2 homologous chromosomes, and this is happening on 2 non-homologous chromosomes, so that's not good either. But that's the 4 different major changes that we can have in a chromosome that can cause problems. And so, again, this is how we get trisomy 21 is actually 2 diagrams ago, that non-disjunction event. And we can see that on a karyotype, that's what this kind of a diagram is called, where we see 3 copies of that 21st chromosome. There are a couple of other trisomies that just, can be inherited. Most of the time when we have trisomies, that results in a spontaneous abortion or a miscarriage is kind of the term that's used for that in more general speaking. But a spontaneous abortion or a miscarriage happens when we have a trisomy up here. And the reason is just because it doesn't tend to be a survivable event. The belief, and we don't have any fact to back this up, but the hypothesis right now is that the reason trisomy 21 is survivable is because it's so small and it codes for relatively few things. Trisomy 18, children with trisomy 18 are usually born, but don't usually live much into childhood. Trisomies of the sex characteristics are actually rather common, or those sex chromosomes are actually rather common, and so you can have extra X's and extra Y's and those kinds of things with the kind of phenotype that usually you wouldn't even notice unless somebody told you. So trisomies with those sex chromosomes are really common, less so with the autosomes, and then certainly very rare the higher up you go in on this diagram. You need to know how to recognize linked genes from data and how to use data to determine distance between genes on a chromosome. All right, so we're going to look at linked genes. And we're going to remember that, whoops, moving the whole thing. All right, we're going to remember that linked genes are 2 genes that are on the same chromosome. So they're physically linked. I can't spell. Chromosome, there we go. They're genes that are physically on the same chromosome connected by covalent bonds. Remember, they're just molecules or macromolecules. So when we're looking at linked genes and the data we're looking at here, remember the goal is to understand how do we use this data to figure out how far away these 2 genes are on the same chromosome. And so this is the diagram that kind of goes you through the process of obtaining data and then what do you do with that data. And so here we have a P generation and an F1 generation. We are not looking at an F2 generation. But from our P generation, we're going to take 2 flies that are homozygous for these 2 traits. We have the body and then the wings. And so here's your wild type with the pluses, and then over here we have the mutants. And we call him a double mutant because he's got a mutation for both of those genes. We take these 2 and we mate them, and lo and behold, we get a whole bunch of wild type flies, meaning that they have both of these traits. Except we now know that they absolutely positively must be heterozygous. Okay, but they're phenotypically identical to this wild type true breeding mom that was up here. So we take one of these now heterozygote flies and we mate them with another fly that is this double mutant. So we have our homozygous recessive, black body, vestigial wings, go through the process for both flies to produce gametes. Well, here, all the gametes are going to be the same. We're going to go through meiosis 1, we're going to go through meiosis 2, and all we're going to get is one gamete because this fly is homozygous. Over here with the female side, life gets a little bit more complicated. So what we have here, presumably, is the wild type chromosome, and then we have the double mutant chromosome. And it'll go through synthesis, and so they'll both be duplicated, and that's what we see here and here. And then we go through meiosis 1 and meiosis 2. But the trick is, during meiosis 1, you have crossing over. Now, we're not worried about crossing over here, not because it doesn't happen. It absolutely is going to happen. But any crossover between these two, or this pair of homologous chromosomes, is identical because those chromosomes are the same. But when we come over here and we're dealing with our heterozygote, if we have crossing over, remember crossing over is occurring between non-sister chromatids, okay, so we have these two are exchanging some genetics. That's what we get here. And you can tell that because we have the light pink here and the red here. So there's some crossing over that's happening. So the crossing over happens, and what we end up with are 4 different kinds of chromosomes from the mom. We have the traditional chromosomes here and here, where there's not been any crossing over. And then we also have these recombinant chromosomes. Now, this whole thing right here is your whole Punnett square. The reason we only have one row is because Dad can only contribute one kind of chromosomes. There's no need for us to pull it out there 4 times. And so we run the cross, we count the babies, and what do we get? We have 965 of them that are wild type gray normal. Okay, so that's like mom. We have 944 of them that are black vestigial. Okay, that's like Dad. Well, then we have 206 and 185 that have one characteristic of mom and one characteristic from Dad. And that is where life gets interesting, because the only way we could have these recombinant offspring, meaning that they have one trait from mom and one trait from Dad, is if crossing over happened and the location of the chiasmata was actually between those 2 genes that were on that chromosome. So the way that we can then figure out how far away 2 genes are on the same chromosome is by counting those recombinants. And that's all that's happening down here. We say, well, we had a total of 2,300 offspring. Of that, 391 are recombinant. It's a 17% recombination. And so in the world of genetics, we just say, okay, well then we're going to turn those percents into map units. Those 2 genes are 17 map units apart. Pretty simple, pretty straightforward. As long as you can keep track of who are your parental phenotypes and which one are your recombinant genotypes, all you have to do is divide in order to figure out your map units. So once you have your map units, you do this, what I showed you on the previous slide, for a variety of different traits, and maybe you do it with, their little, they call them aristae, but the little head whisker things, and you do it with body, but then you do the whiskers and the red eyes, or, you do a combination 2 at a time for all these different phenotypes, and you figure out all these map units, and you're able to start building a chromosome map. And the chromosome map is just saying, okay, how far apart are all these different genes relative to each other? What this works under the assumption of is that crossing over is equally likely at each spot on the chromosome. But if that is in fact the case, then we're able to get these numbers that represent the percentage of time when we allow mating to happen that we're going to expect crossing over to happen. If we have a number bigger than 100, that means even though these genes, these alleles, or genes rather, are physically on the same chromosome, crossing over is virtually guaranteed to happen at some point on that length. So even though they are physically linked, for all practical purposes, those 2 genes would not be linked. All right, last little slide about recombinant versus parental type offspring. This is from textbook 232, or 236 rather. And so this experiment with the flies is actually not altogether too different from one of the experiments that Mendel did, where what he did is he took 2 parents, okay, and was able to get recombinant offspring as well. Same thing, he just didn't do the math and didn't understand the chromosomes. So don't get too addicted to seeing flies in all of these examples when we're dealing with linkage and sex-linked and all that kind of stuff. this is not organism specific, so be ready to get this information from peas or dogs or any kind of fictitious organism that they might decide to throw at you on your AP test in May. So that's it. Thank you. I hope your quiz goes well, and I'll see you later.